Welcome to another screencast where we'll be discussing regulation of gene expression in prokaryotes, and we'll be talking this time about the operon in general and then a specific example of the operon called the Lac Operon. So by the end of this, hopefully you will be able to describe the parts of an operon, and there are three parts to them. Describe the role of the regulatory gene, describe the role of the repressor protein, and describe the function then of the Lac Operon. Right? So regulatory gene and repressor protein here and here could sometimes get these two things confused, so please do slow down your thinking process at that point. And again, by the end, why the Lac Operon, be able to explain why the Lac Operon is said to be an inducible operon. So we'll dive into this here. Prokaryotic cells on the left, eukaryotic cells on the right. We have our gene expression. Again, transcription followed immediately by translation in prokaryotes. In a eukaryote, we have transcription followed by some sort of post-transcriptional modifications, and then translation out in the cytoplasm. So the regulation here in the eukaryotes is addressed in separate screencasts. Here we're talking about how prokaryotes are going to be regulating the expression of their genes, that is, via an operon model. So an operon is the way prokaryotes regulate gene expression, is one way. It's the most common way, one that you are responsible for being able to describe and explain. So there are some parts to this operon. So first I want to identify here's a DNA sequence. So this is a region of the genome of a prokaryote. This part right here is the regulatory gene. So this would be a regulatory gene, a sequence of DNA. This regulatory gene then codes for an mRNA molecule, which then when it is translated, so here's transcribed and translated, gets us a repressor protein. Repressor protein. Okay, so all of this that I just described is separate from the operon. This is not part of the operon. This is upstream from the operon. This is, so you see there's a break in the DNA here. So this is not technically part of the operon, but will play a role in how the regulation is happening. So this regulatory gene codes for a repressor protein. Right? These repressor proteins come in different flavors. Right? They can be made in an inactive form, or they can be made in an active form or conformation. Right? And reference a previous screencast there, again, where we're talking about active enzymes, inactive enzymes. Right? So this repressor protein is an enzyme. It's not really an enzyme, but it has this repressor nature to it. And it's going to have this is going to be the allosteric site, where this is then the active site. So this is the allosteric site, and this is the active site. Okay? So the name of this protein sort of tips you off as to what it might be doing. Repressing. A repressor is something that slows down or stops something else. Okay? So more on that later. So there's the regulatory gene and the repressor protein. Now to come to the operon proper, the operon proper is a sequence of DNA. Right? It's from here to here. And that sequence of DNA has a promoter region. And this promoter region, this promoter right here is where the RNA polymerase would bind. So this is where we see RNA polymerase binding. And then there's another regulatory sequence right here called the operator. And this operator and this promoter, again, these are both sequences of DNA. They don't code for anything, but these are regulatory sequences. So if you were ever asked to describe a regulatory sequence, you could use a promoter and an operator as examples of a regulatory sequence. So these operators, these promoters, the things are going to bind to them. RNA polymerase binds to the promoter. And the operator, what binds to the operator is a repressor protein. A repressor protein binds to the operator, as long as that repressor protein is active. If it's inactive, then it won't bind to the operator sequence. Okay? The third part, so this is one part. You have the promoter, you have the operator, part of the operon, and then you have everything that's downstream from that, and these are the genes of the operon. And these are also sometimes referred to just as the structural genes. Structural genes or genes of the operon. These structural genes are genes of the operon, so if we call this like gene A, gene B, gene C, gene D, and gene E, right? So these are several genes that are all clustered together, a family of genes that when they are transcribed, so there's no arrow here showing this, but you see this RNA polymerase, if it's moved from the promoter down, you've then made this piece of mRNA, so you've done transcription. And then you end up getting polypeptide here expressed from that. So polypeptide A, polypeptide B, polypeptide C, polypeptide D, polypeptide E, right, are made sort of all at the same time here. So prokaryotes a lot more efficient in terms of genome or genomic material, right? So all these enzymes here, probably these polypeptides probably all play a similar role in a metabolic pathway. Okay. So again, the three parts of the operon: the promoter, the operator, and then the structural genes of the operon. Okay, or they are sometimes referred to just as the structural genes. Right? So let's look at an example and see how this is going to work. The classic example is called the Lac Operon. This Lac is short for lactose. Right? And so lactose is a sugar. It's a disaccharide. And that disaccharide, excuse me, that disaccharide lactose can be broken down into glucose is a monosaccharide, and galactose is another monosaccharide. And then this glucose and this galactose can be used in cell respiration metabolically. Okay? So that glucose, that galactose can enter glycolysis, and then the whole thing goes in there. So lactose cannot go right into cell respiration. So what has to happen is you need an enzyme, right, or a series of enzymes that will break down lactose into glucose and galactose. Right? So those enzymes are, of course, encoded by genes. So when a bacterium is given lactose as an energy source, as a nutrient, it takes in that lactose, and that lactose will then be a signal. Will be a signal to begin the expression of genes that, when they are coded, code for the enzymes that break down lactose. Okay, sorry. So let's go up here and look at our diagram. Here's our DNA. So this LAC-I right here, this is the regulatory gene. When it's transcribed and when it's translated, yields a repressor protein that is in an active conformation. So this repressor protein right here is made as an active conformation. So the expression of this gene is constant, right? So this is always going to be on. Always on. And just as a side note, when I say on, I mean that gene is being expressed, is being actively transcribed, and is actively being translated. Right? So always on, this is happening. You're making active repressor protein. And so if you had this active repressor protein, this repressor protein would bind up here to the operator. Right? And if you bind that to the operator, then this RNA polymerase physically cannot move down the structural genes of the operon. Right? And so these structural genes of the operon right here, this LAC-Z, LAC-Y, and LAC-A, all code for enzymes, respectively, beta-galactosidase, permease, and transacetylase. Right? Suffice it to say that these enzymes here, right, you don't need to memorize the names of each of these, but beta-galactosidase, permease, and transacetylase will all play roles in breaking down lactose into glucose and galactose. Right? So the hydrolysis of lactose here is mediated by these enzymes and the taking up of that lactose, etc. Right? So the cell is not going to want to, this is anthropomorphizing it, the cell doesn't want to express lac Z, lac Y, and lac A when there's no lactose present, because then you're just wasting energy doing this transcription and doing the translation. You're wasting resources doing that transcription, translation. You're making enzymes that you aren't going to do anything because lactose isn't present. So the way this works is that if lactose is present, right, and we're going to call this, and this allolactose is a what do you call that? Different structural form. I'm having a complete brain meltdown at the moment. Same structure, but different molecular formula, and not an isotope. But it's a different form of that lactose. So this lactose right here acts as an inducer. Now, this lactose, when it's present, this allolactose, when it's present, will bind to this part. And again, this is the allosteric site. And when allolactose binds to the allosteric site of this repressor protein, it turns that repressor protein into an inactive repressor. So this inactive repressor now cannot bind to the operator up here. And so no binding to the operator does not bind operator. And if that is the case, then RNA polymerase is allowed to then move down here and express or transcribe lac Z, transcribe lac Y, and transcribe lac A, and then into this mRNA, which is then translated and translated and translated, and you get these enzymes, and then that breaks down lactose into glucose and galactose. So when that happens, if lactose concentrations go down, you would then expect to see this allolactose not binding to the allosteric site as frequently, and you'd expect to see more of this form of the repressor protein. And then when it's in that form, that repressor protein can bind up here to the operator and stop that RNA polymerase from moving down the structural genes of the operon. Right? There's an animation right here. I would recommend clicking on that on the PowerPoint and going and watching that tutorial as well. So, a couple things here. If you followed that, then you should be able to kind of do this, right? Tell me what would go right here, right? And you're going to follow this pattern right here. So over time, right, what would we be measuring there? And what would be the stimulus to get this to come back down? And what's the stimulus to get this to come back up, right? So again, what is this and what are the stimuli? So that's something to kind of work on and discuss and bring that to class with you. Also, here we go. If I give you this blank diagram right here, first, can you label all the pieces and the parts? So that is identifying all the pieces and the parts of the operon. And then describe this. So answer this question right here. The Lac Operon shown below, is it on or is it off in this diagram? And then explain how you know. Describe the conditions which would make it so, the conditions of the cell, inside the cell, outside the cell. What conditions, what would the stimulus be? Right? So there's something to practice. And then here's another similar diagram but different. So that Lac Operon, is it on or is it off? Explain how you know. And then describe the conditions which would make it like that. Okay? So you should be able now to describe the parts of the operon, describe the role of the regulatory gene and the repressor protein, and then describe the functioning of the Lac Operon. And then is this Lac Operon, is it an inducible operon or is it a repressible operon? Well, I'll tell you that it is an inducible operon, right? But why? Why does this make sense, right? If you're going to induce something, how do you get that to happen, okay? So thanks for listening. Bring your questions, confusions, clarifications to class, and practice those, practice identifying there on those blanks. Okay.